Okay, in this lecture I will first discuss the concept of biocompatibility, and I will give you some definitions, useful definitions which are related to biocompatibility and host response, and I will also discuss that what are the different property requirements for a given synthetic material before the biomedical application can be realized. So, biomaterial, the way it is defined, it is as that any natural or synthetic material that interfaces with living tissues. So, I highlight here the word synthetic material, because synthetic material means it is a bone replacement material, and this material is fabricated outside the body environment in laboratory or in industry or in research labs and so on, and this material does not contain any biological substances like cells, proteins, etc., and this is purely synthetic material. It can be organic material like polymer based material, or it can be inorganic based materials like metals or ceramic based materials. And some of the important things in this definition that you need to follow is that it should have a good interface with living tissues. That means the synthetic materials, it is expected that when it is implanted inside the body environment, that it will establish, or it will have good interface with living tissues. Now these tissues, these are nothing but actually self organization of multiple type of cells. So you can note down these words like this is self organization of different cell types in a particular fashion, and then this is called biological tissues. So therefore a synthetic material, when it will have interface with tissues, in other words it will have interface with the self organized layers of different cell types, or biological fluid. Biological fluid can be serum, biological fluid can be blood, biological fluid can be blood, serum, and etc., and it should elicit desired biological response. Now the way desired biological response means it can be different for different type of biomaterials. Just to give an example, when you are talking about total hip replacement, it is supposed to bear the load. When you talk about the heart valves, then it is supposed to facilitate the blood flow in the heart. So artificial heart valve, for that mechanical property is not important, but total hip replacement, for that mechanical property is also important after the biocompatibility property. The second thing is that any artificial bone replacement material, that should have a good cell adhesion property and tissue formation property. But when you talk about the artificial heart valve, then it should have a property which should not allow blood cells to adhere on that artificial heart valve, so that blood cells cannot coagulate, blood cell cannot form any thrombus on that heart valve surface. So you understand. So biocompatibility is a broad term, and then it is also application specific. So for bone replacement materials or total hip replacement, whatever you call it, biocompatibility, that kind of concept is not valid when you go to the artificial heart valve applications. Okay. So this thing should be taken into consideration when you design a particular biomaterial. Now second important definition is that biocompatibility, that is the ability of a material to perform with an appropriate host response. Now what is host here? Whenever we talk about host, host is the human patient. So host response and host, whenever you talk about host, means it is a human patient or any animal, for example. And appropriate host response means like this host will not suffer some kind of inflammation just because of the implantation of the material. So that is what is important, and in a specific application. Now what is the host response? The response of the host organism, local or systemic, to the implanted material or device. Now host organism means in the knee joint applications, it is the tissues around the knee, or if it is hip joint, that is the tissues around the thigh region of a human patient. That is important. How those tissues will react to the implanted material, that is what is known as the host response. So now you have learned three important definitions. Number one, what is meant by biomaterial. Number two, what is meant by biocompatibility. And number three, what is meant by host response. Now this definition of biocompatibility is more detailed, and I am now giving you a more recent definition that is proposed by David Williams, who is currently the editor-in-chief of the Journal of Biomaterials. So the Biomaterials, that is the journal, and David Williams is the editor-in-chief. Now, what he proposed, the definition of biomaterials is that biocompatibility refers to the ability of a biomaterial to perform its desired function with respect to a medical therapy. Medical therapy means like in a particular application that you are developing this biomaterial for, without eliciting any undesirable local or systemic effects in the recipient or beneficiary of that therapy. Who is the recipient or beneficiary? That means it is the host, that is the human patient. But generating the most appropriate beneficial cellular or tissue response in that specific situation. Means most appropriate beneficial cellular tissue response means whatever is required, like for bone replacement you want that blood tissues or tissues to grow inside the material, or any other material, bioresorbable material means like you want the material to dissolve and degrade with time in vivo condition, and it should be replaced by the natural tissues which is surrounding that material. So what he is saying is that it should have a good cellular response or tissue response in a specific situation and optimizing the clinically relevant performance of that therapy. That is what I was trying to mentioning to you a few minutes ago, that whatever biocompatibility property you require for heart valve application is different from whatever biocompatibility property that you require for hip replacement or knee replacement, for example. Okay. So these two things should be very clear for any for many students who pursue the research on biomaterials. They always think biocompatibility means it is always the cell adhesion test, MTT, cell viability, but that is important for orthopedic applications, but not for cardiovascular application. Cardiovascular application means like heart valves that I mentioned. For that, different type of test is required, that is called blood compatibility. So it is not biocompatibility, but it is again blood compatibility test is required. So now you understand roughly that what is the concept of biocompatibility and why the biocompatibility concept is different for different applications. Okay, now here it is been little bit more defined or described that that ability of a material to perform that compatibility or harmony of the biomaterial with the living systems. Living system means that is the human being system. Now there are two types of compatibility. What they have defined here, one is the surface compatibility and one is the structure compatibility. Now surface compatibility means like, now let's go back to the example that I have given you in the earlier lecture, like where I have discussed the total hip replacement. You have the titanium stem and on the top of it you have this hydroxyapatite surface coating. So the bulk composition is titanium, surface composition is hydroxyapatite. So what I am meaning here by surface compatibility is that it is not the biocompatibility property of titanium here is not important, but it is important whatever hydroxyapatite coating you are putting on the surface of titanium. That is what I am trying to say here. So that is the chemical, physical or biological suitability of the implant surface to that host tissue, that is important. So here host tissue will not see titanium directly. Here host tissue will first experience the hydroxyapatite coatings before it will see the titanium substrate. The second one is the structure compatibility. Structure compatibility means that is the optimal adaptation to the mechanical behavior of the host tissue. Mechanical behavior means like what is the response, mechanical response of the tissue or the bone surrounding that. That is what I was mentioning in the last lecture, that you have the elastic modulus of the cortical bone that is 3 to 80 gigapascal. Now if your elastic modulus of the implant is too high, then implant will bear most of the load, not the bone, and as a result your implant will be detached from the surrounding bone. So that is what it is important, that your optimal adaptation to the mechanical behavior of the host tissue or host bone. Bone is nothing but a hard tissue. So this bone, how this is optimally adapted as far as the mechanical properties is concerned, that is what is meant by structure compatibility. Now you see that there are two things, one is the surface compatibility and one is structure compatibility. Structure compatibility mostly means for the load bearing implants how mechanical adaptation process is possible. Okay, now here is the biocompatibility interactions. What I have shown here, that one is your biomaterial and one is the material function. Material function can be your two things. One is that biological function, biological properties. Biological properties means what is the that how the cells will grow on the surface or how this bacteria will be adhering on the surface, etc. and you have the mechanical properties also, like what is the elastic modulus, strength, etc. And then one is the patient. Patient is your human patient. So patient is your living tissues or living organs. So in biological terms, you can replace the patient by living tissues or living organs. Material function means that is your biological property, mechanical properties. Your biomaterial means it is your synthetic material, right? And this synthetic material you are actually fabricating outside the human body. And this synthetic material, what is the manufacturing processes, that I have explained to you in the last lecture. That is, for metals it is the standard casting, rolling, etc. For ceramics it is more powder based methods like processing, like powder processing, sintering, and so on. So now there is the intersection of these three, and this intersection region is this one, what I have been now shaded. Now these intersections is nothing but what is your biocompatibility property is important, where biomaterial, it should have a suitable composition, it should have a suitable or desired material function, like in terms of biological properties and mechanical properties, and that should match with the specific tissues or specific biomaterials organs where you are implanting this biomaterial into the human patient. You understand? So that is the common area or common subset of these three actually will reflect in the biocompatibility property. So I am giving you all these slides so that you can have a fairly good understanding of this, what is meant by the biocompatibility property. But in your laboratory scale experiments, you do not use any patient things, right? So that's why it is called in vitro. In vitro experiments means like it is a simulated environment of the patient's one. So as I said that what is in vitro and what is in vivo. Now typically in literature you will find that there are a lot of in vitro experiments, and in vivo experiments are not that significant in numbers compared to in vitro experiments, because in vivo experiments that require ethical committee clearance and the animals, which is not very easy to handle and so on. So in vitro experiments, the way it is defined as that it is the laboratory scale simulated experiments. Now what is meant by simulated? You are essentially trying to simulate as close as the environment possible which is prevailing in the human patient or human body. So therefore, as I have explained to you in the first lecture, that human body, that core temperature is 37 degrees Celsius, the pH of the blood is maintained as 7.4, and therefore most of the in vitro experiments that you carry out, so whether it is cell culture, whether it is the in vitro dissolution, everything, there the temperature is always maintained at 37 degrees Celsius, pH is always maintained at 7.4. And to give you further details, like cell culture experiments, they are maintained at 5% CO2 level and 95% air. Why? Because that also simulates exactly that CO2 level inside the human body. So human body, if the CO2 level is more than 5%, that means your human organs are not functioning well. That's why CO2 level has increased. So that is the optimum level of CO2 that is always available inside the human body, and that is the reason you require 5% CO2 in your cell culture experiments, right? So laboratory scale experiments, no animal involved, and also these in vitro experiments, the other thing is that it is very rapid and mostly initial screening experiments, because animal means you are sacrificing some animals for your in vivo experiments, and that is not good, right? So you should first do the in vitro experiments, you select the best material out of this in vitro experiments, then you do in vivo experiments. That is the logical sequence of doing the biological testing. It is inexpensive in vitro experiments, and it is poor representation of physiological conditions. Why it is poor representation? Because I have mentioned in the last lecture that in a human body environment, your pH of 7.4 is only maintained at blood, but that pH also changes depending on whether it's urine, whether it is gastric environment or gastric fluid, etc., etc. So pH 7.4 is not constantly maintained all the time inside the human environment. And second thing is that temperature 37 degrees Celsius is also not maintained throughout the human body. So again, that is also grossly poor representation of the physiological environment. Third point is that Normally all human tissues, they are actually aggregates of different cell types, but your cell culture or bacteria culture, you are always using only one cell line. You are not using multiple cell lines together during your cell culture or bacteria culture experiments, right? So all these things actually shows you that it is a poor representation of the physiological conditions. However, it is always a good as a first step. Then in vivo experiments, like animal experiments. Animal experiments means like you can use rabbit, you can use rat, you can use mouse before you can go for the human trials. Now you always start with the smaller animal. Smaller animal means like rat. Then you go to little bigger element like rabbit. Then you can go for the largest element is called, largest animal is called human being. Now why I am putting this stress on the small animal to intermediate animal to large animal, because if you talk to any clinician or medical practitioner, they always tell you that rabbit model experiments. Rabbit model experiment means that is the intermediate animal model of experiments that you are doing in the rabbit, in case of rabbit, because smaller the animal, lesser the complexity of the body environment in terms of pH variation, in terms of temperature variation, and so on. Larger the animal, larger will be the volume, larger will be the variation, because the larger the animal, more the volume of the body that you are dealing. The smaller the animal, it is the smaller the volume of the body environment that you are dealing with. So the variation will not be that significant when you are talking with the help of the human being. So that is what is meant by, that is the different in vivo animal models that you really want to use. Now it has a demanding protocols like Animal Welfare Act you have to follow. You have to also get the clearance of the ethical committee and so on. The right animal model actually approximate human environment, and it is the second step prior to the clinical use. Clinical use means like in the human beings. Now in vivo testing, in vitro tests cannot replace in vivo tests. Why? Because in the in vitro test you cannot simulate that whether there is any inflammation or not. Inflammation can only be realized when you put the material inside the human body or inside the any animal. Okay. Second one, there is no immune response. Immune response means you are not dealing with any immune system. Immune system means that is inside the human body or inside the any animal. So there is, you cannot study any immunohistological response of any material. Third point that I already mentioned, that in vitro test always deals with the single cell type. Let's say it is a connective tissue cells like fibroblast. If it is a bone cell type, that is osteoblast cells. But it does not involve multiple cell types in the single experiments. That is what is missing in the in vitro experiments. Number fourth is that there is no tissue remodeling. Tissue remodeling means, I will come back to this later when I will discuss little bit of the tissue engineering concept. Tissue remodeling means you are not giving enough environment or enough biological environment so that there is a ECM, that is extracellular matrix formation, and there is remodeling of the extracellular matrix composition all the time, dynamic. So no dynamic changes in the extracellular matrix that is possible in the in vitro experiments. Now in vivo test, whereas provides interactions of the different cell types that I have already mentioned, effects of hormonal factors, interactions with extracellular matrix, that is not possible in the case of in vitro test, interaction with blood-borne cells, proteins, and different biological molecules. All these things makes in vivo experiments much more meaningful and much more complicated. Okay. So in summary, I must mention here that whenever you do the in vitro test, you cannot definitely say on the basis of your in vitro test that yes, this material will be extremely suitable for in vivo applications, because your in vitro test can be very positive, but that does not ensure that your in vivo test also will be positive, because in the in vivo test you have some additional parameters which will be involved, and that will significantly influence the biological performance or biological properties of any biomaterial. Okay. So the underlying theme, this is the very final level understanding, that your in vitro test only can say that in these simulated environments this material has good biological properties. It does not ensure that this material will be clinically successful for some load bearing implants or some from heart valves and so on. So for that you need to use the suitable in vivo test in a particular animal model, and you can start with the small animal to go to intermediate animal, go to the large animal model experiment. Now general property requirements of the implant materials, as I have been mentioning right from the beginning, the biocompatibility is the most important property that a material must have before it can be used for biomedical applications. Now biocompatibility property, you now have broadly understood that what is meant by biocompatibility, what is the textbook definition of biocompatibility, how it is properly defined. The second one is that it should have extremely high corrosion and wear resistant. Now this is more appropriate for load bearing implants, because in the load bearing implants your material also many times experience some reciprocatory motion, like in the acetabular cup, femoral ball, those kind of combinations which I have mentioned for THR or total knee replacement. So total knee replacement or total hip replacement, your wear resistance is also important. Third one, as I said, elastic modulus. So elastic modulus matching with the neighboring bone or cortical bone, that is important. Fourth one is that high fatigue and compressive strength, again I have told you for load bearing implants. So fatigue strength I have shown you. Remember that fatigue strength in the, if you do the normal laboratory environments without seawater, you can measure high fatigue strength, but when you use the sodium chloride rich seawater kind of environment, then you are bound to have much lower fatigue strength compared to that in air. So again the material which experiences very high fatigue strength in air, that should not be the design criteria. Design criteria should be what would be the fatigue strength in the simulated body fluid environment. Last one is the density. I also mentioned the density just to give an example or to refresh your memory, that steel based material that has a density of 7.8, whereas alumina is a density of 3.9, which is just half that of the steel. So therefore same volume of implant will weigh less if we use ceramic compared to metals, and if the weight is less, the patient will be feeling more comfortable for the same implant if it performs well. This is the kind of a snapshot view of that what are the property requirements for biomaterials. Now here I have mentioned that elastic modulus is around 30 to 80 gigapascal. This is depending on what is the kind of specific applications. Now density is 3 to 4 gram per cc, that is more relevant for ceramic based materials. Fracture toughness, it should be more than 3 MPa square root meter, because if you remember your cortical bone, the fracture toughness that varies around 2 to 12 MPa square root meter. So if it is more than 3 MPa square root meter, it is better. Fracture strength, it should be as high as possible so that it can be used for load bearing application like 200 MPa. Hardness should be more than 4 gigapascal, because the typically bone are soft and bone does not have very good very high hardness property. Now in vitro property, first one, it should be non-cytotoxic. Non-cytotoxic means cyto means cell, toxic means it should not have any undesirable toxic effect. So it should not have any cytotoxic property. That means that cells should survive well when you will sit these cells on this material surface. Then second point is the cell adhesion, like fibroblast or osteoblast cells. Now the type of cells also depend on what kind of applications that you will be using. For example, fibroblast cells are the most connective tissue cells, and these connective tissue cells are actually the cell lines which comes to the wound site very fast, very promptly before the osteoblast cell lines. Osteoblast cells are the bone forming cells. Osteo means bone, osteoblast means bone forming cells. Fibroblast means, again fibro means it's the cells of the fibrous tissue or the connective tissue. So that way you can remember these typical biological names. Third one is that calcium phosphate rich layer formation in SBF. Like whenever you do any in vitro dissolution experiments, you first check whether this biomaterial is capable of forming any calcium phosphate rich layer formation. Irrespective of the composition, if that biomaterial can have calcium phosphate rich layer formation, then what will have? It will have very good osteological, or it will have very good biological response when you put it in vivo conditions, because in vivo conditions also it will first form calcium phosphate rich layer formation. If it is calcium phosphate rich layer formation, then what the neighboring tissues or bones will feel, because each bone also will have 65% calcium phosphate. So the bone will find as if that similar composition is existing next to the synthetic material, and therefore it will have good bonding or good biological bonding with between the tissue or bone along with the synthetic biomaterial. Fourth one is that hemocompatibility. Hemo means blood compatibility, but again that is only applicable for the heart valve applications, not for other type of applications. It is not the primary criteria. In vivo property means this should have a biomineralized layer or bone like layer formation. It should have a biological interface or bone apposition, and it should have a controlled degradability. So I will show you later that when a biomaterial you implant inside the human body, then this biomaterial should have good bone formation or neobone. Neobone means that is new type of bone which was not existing before you put the implant in the human body. So if this neobone is formed, then it should have a good in vivo biocompatibility property. So like this is like overall view of the different property requirements of the biomaterial. This is like more details about the what is the different bone analog materials. Now the property requirement for different bone analog materials, it can be, so there are 3 plus 2 5 plus 3 8 circles surrounding this. So that means 8 different aspects you need to take care when you are actually developing bone analog material. Bone analog means it is like bone replacement materials. You are developing materials for using as a like a natural bone. First one is a processing. Now processing means it should have a good processability to make complex shapes. Remember, bone does not have a very simple shapes like rod like shape or typical circular type shape, but bone can have a very complex shape. So whatever material that you are producing in the laboratory scale, you should be able to produce it in a much more complex shape than simple shape. Second one, CAD/CAM based designing to produce desired shapes and porosity. CAD/CAM means computer aided design and computer aided manufacturing. So this computer aided designing, you can develop the materials with controlled porosity using the CAD code, that computer aided design code, or computer aided manufacturing process. Third one, whenever you use the hydroxyapatite coating, you should have an optimum thickness, and it should have a good substrate coating interface and integrity. That must be ensured before good processing, before this material can be used in real applications. When it comes to microstructure, it is important that whether you have a presence of a biocompatible or non-toxic phases, because if you have alpha beta gamma phase, if one of the alpha beta gamma phase has a toxicity effect, then the entire material will have a cytotoxic effect. So you cannot use this material in real applications. Then second one is a microporosity. Microporosity means any pores which is less than 10 micron, and macroporosity means any pores which is in the range of 40 to 100 micron. So if it is less than 10 micron porosity, then it is very good for initial cell adhesion property. If it is greater than 40 to 100 micron porosity, this is macroporous, then it is very good for nutrition and nutrients and blood channels, everything to enter, and tissue formation and tissue ingrowth also to take place. Third one, for the hydroxyapatite based materials, it is important to have a combination of hydroxyapatite and TCP phase. What we call is that BCP, that is a bicalcium phosphate phase, and this bicalcium phosphate phase is important for these hydroxyapatite based materials. Now coming to the physical properties means lower density that I have mentioned. Elastic modulus is 70 to 80 gigapascal, that I have mentioned why it is required. For many load bearing applications, compressive strength is important as well as flexural strength is also important. Fracture toughness means it is the resistance to the crack growth, and this resistance to the crack growth should be as high as 3 MPa square root meter or more. Now coming to the other properties is like electrical properties. Now the question is that why electrical properties is important. Bone itself is a piezoelectric composite of the organic and inorganic phase, and piezoelectric composite means this bone analog material should have a dielectric constant which is around 1000 or more. it should have electrical conductivity property, which should have a 0.04 ohm inverse per meter, and it should have a piezoelectric coefficient which is somewhere around 0.5 to 0.7. So this combination of properties, if it is ensured in these synthetic biomaterials, then what will happen? This biomaterial would be an ideal bone material. Now this 4 is mostly from the physical and electrical and functional and mechanical properties as well as the processing. Now these 4 in vitro properties, antimicrobial properties, in vitro properties is the bio oriented properties. Now these are all like physical and biological physical properties mostly, as well as the processing. Now in the biological properties, first point to note here is that in vitro biomineralization. In vitro biomineralization means, first of all, in vitro means laboratory scale simulated environment. Biomineralization means it should ideally be able to form apatite layer at the early stage of incubation in SBF. Many times some biomaterials, they take 4 weeks before they can form calcium phosphate rich layer formation. There are 2 or 3 type of materials, let's say A, B, C. Some material they take 1 week form that calcium phosphate rich layer formation, another material they take 3 weeks, another material they take 6 weeks. But the first material which it takes 1 week for the formation of calcium phosphate rich layer formation, that material is highly biocompatible compared to other 2 materials. There is no way that you can quantify, or there is any quantification index or how good biomaterial is bioactive or not, but this is the way you can roughly find out that how you can evaluate the biomedical potential of a good biomaterial. Then second one is the less leaching of ions, like if the ions are leached and if the ions can cause some toxic effect, that is also should be avoided. So therefore that less leaching of ions should be preferred. Coming to the other part of the biological properties is called in vitro biocompatibility property. Like first one is the cellular functionality. Cellular functionality means it is a better expression of MTT, which is a cellular viability property, or ALP, that is the osteogenic differentiation or osteoblast differentiation, and osteocalcin, that is the bone mineralization property. Now all these assays I will be dealing with it much later in more details. Now there is a good cell fate processes, like whenever you will seed the cells on the biomaterial, it should be able to migrate, cells should be able to migrate, shells should be able to differentiate on the contact with biomaterial, shells should be able to proliferate also. So all this is called in combination, like proliferation, migration, or adhesion, or differentiation. All these properties together is called cell fate processes. So this is like a biological term. Cell fate is nothing but combination of proliferation, then migration, differentiation, and cell adhesion. Next one is the antimicrobial properties. Last lecture I have mentioned that hydroxyapatite is the most biocompatible material or most bioactive material, but the problem with hydroxyapatite is that it does not have any antimicrobial property. Antimicrobial property means whenever you seed bacteria cells on this hydroxyapatite surface, then what will happen? These bacteria cells can easily adhere and they can easily survive on the biomaterial surface. So that can cause infection in the whenever you are implanting in the human body. But if you add some silver to hydroxyapatite, they should be able to kill all the bacteria. So you get a material with antimicrobial property. Third one is that in vivo biocompatibility property, like it should not cause any inflammation, it should ideally cause new bone formation, that is the new bone that is formed during the early stage of implantation. And third one is the good integrity of the natural bone or implant surface. So that integrity means there should not be any gap. Suppose this is your biomaterial and this is your human bone. So this interface should be continuous. There should not be any gap between the biomaterial as well as the natural bone. So this is what I meant by good structural integrity at the natural bone implant interface. Okay, as I said that biological tissues, they contain self-organized aggregates of the multiple cell types and a large number of cells in a particular fashion. So therefore it is important to first understand the properties of the cells and what is meant by cell, biological cell. Now here I am giving you a definition. Biological cell is a self-contained unit which is capable of duplicating itself given the proper nutrients and environment. and in general cells are enclosed by lipid bilayer. So that is important because cell membranes, they have a lipid bilayer and they contain the necessary genetic material. Now this necessary genetic material, it is enclosed in the nucleus. So that is the DNA, RNA, right? And this genetic material can direct the continued propagation of the cell. Continued propagation of the cell means the cell will go through a cycle, right? Cell will differentiate, then cell will go to the death or cell necrosis and so on. So that entire cell cycle is also governed by that, how this genetic material they will perform. So there are two things here you can find out, that cell membrane, they contain lipid bilayer, and cell is a self-contained unit which is capable of duplicating itself because it contains a necessary genetic material. First let us see that how this plant cell and animal cell they look like. Now this plant cell typically has a very regular type of structure, whereas animal cell it does not have any regular structure, it is more like irregular structure. This you are seeing like a thin section of an animal cell. Now it also has a nucleus, and nucleus is enclosed by a nuclear membrane. You have the cytoplasm which is inside the cell. You have the mitochondria, you have the Golgi body, all those things you can see in the next slide. This is the molecular biology of the cell. So you have the lipid bilayer, that is the cell membrane, right? It is contained by the cell membrane, which is nothing but a lipid bilayer here. And this lipid bilayer also has a particular properties that I will come later. Now this entire entire cell, it is the matrix of the cell, which you can call as a matrix, which is called the cytoplasm. So essentially cytoplasm means that it is actually the entire matrix of the cell. Then you have the one of the important organelles is the nucleus. Now nucleus again is enclosed by nuclear envelope, and it is a nucleus, and you have the entire material which is inside the nucleus is called nucleoplasm. Now this nucleus, I will come back to later, that nucleus also has a nuclear pore, or that means this membrane, whatever you are saying, it is not a very compact layer, it has also some pores inside the nucleus. There are some of the important organelles of this inside the cells which are called cellular organelles. For example, this is called microtubules. So these microtubules are contained in the cytoplasm. So it is one of the component of the cytoplasm. Other component of the cytoplasm is called actin filaments. So these are like called actin filaments here. Then you have the ribosome. Then what is the called protein synthesis. Then you have called Golgi apparatus. Now this Golgi apparatus is as close to the cell as possible, and you have the large number of mitochondria here, and mitochondria is called the powerhouse of the cell, right? Because it is used in the ATP synthesis, and ATP synthesis will lead to the more energy that the cell requires for its survival. What are the other things that you have see? That you have the intermediate filaments, you have the actin filaments, and you have the this is intermediate filaments, this is the part of the cytoplasm. You have the actin filaments, that is also part of the cytoplasm. Now there is the other things, these organelles, these irregular shaped organelles, these are called ER, which is called as the endoplasmic reticulum, okay? And you have the mitochondrion here, you have this chromatin that is contained, that is nothing but DNA which is inside. So you have the three components of the cytoplasm. Let me summarize. These components, one is the microtubule, actin filaments, and intermediate filaments, okay? You have the other important thing is that called Golgi apparatus. Then other important cellular organ is called endoplasmic reticulum. From the energy point of view, it is important to have the mitochondria in the material. And the outside you can see that is called extracellular matrix, what is known as the ECM. It is designated as the ECM. Because many times later on I will refer this ECM, and ECM actually essentially means it is extracellular matrix. This is also another cell type. I mean, for different cell type, the cellular surface or cell size or the cell morphology is also different. It does not necessarily mean that all the cells will have this kind of unique size or shape. And this size you can see, this is like 5 micron. This is the size. So the entire dimension of these cells can be as high as 100 micron or little higher than 100 micron. Okay. Now here you can see this is the more kind of regular circular type of cell morphology, and what is more important, you can see that the cell membrane which contains this lipid bilayer, this you can clearly see here. Now again you can see more clearly the Golgi apparatus, you can see the mitochondria in this here, and also endoplasmic reticulum which is surrounding the cellular nucleus, and you have the nucleoplasm here inside the nucleus. So nucleus contains the genetic material that is required for the gene related activity like DNA, RNA, etc. Now those you have also studied in your school biology, but this is important to recall all the important features of the cell biology here. Now other things is that major intracellular compartments, they are kind of separated by permeable membranes. Now if you go back to this particular view graph, or here you can see mitochondria. If you look at the mitochondria morphology here, then mitochondria is also contained by a envelope which differentiates from the cytoplasm here. So each mitochondria you can see a membrane, just like a cell is enclosed by a membrane. Similarly, all these cellular organelles also are enclosed by some membrane. Also, if you see here the Golgi apparatus and nucleus, that is another important cellular organelles, and this nucleus is also enclosed by a nucleus membrane. So that is also another membrane. So that is what is meant by here, that major intracellular compartments, they are separated by a permeable membrane. And this has been shown here, like this is a plasma membrane, this is a nucleus, and this is a ER, that is the endoplasmic reticulum. This is, you have a mitochondria, and you have the endosomes here, which are also some small elements. You have the cytosol. Cyto means cell, sol means it is like a matrix of the cell. You have the Golgi apparatus, you have endoplasmic reticulum with membrane bound polyribosomes, you have the nucleus. Okay, this is the structure of the cytoskeleton in eukaryotic cells, and this cytoskeleton, it has a different organelles, as well as this entire structure of the cytoskeleton, it has three different components. As I said, that it contains the actin filaments, then you have the intermediate filaments, and then third one, if you go back to this slide, I have already mentioned that it is the microtubules. These three components, these actually microtubules is the third component. These three components, they contained in the cytoskeleton. Now here it shows that how these various organelles, they actually look like in reality. So this is like a more like a shoe type of morphology in the mitochondria, and it is the powerhouse of the cell. You have the rRNA synthesis region, and you have the nuclear envelope. Now nuclear envelope, you can see these contains lot of pores. Either it can be macropores, it can be micropores. You have the smooth endoplasmic reticulum. It has a very complex type of structure which is also there, and you have a rough endoplasmic reticulum which does not have a very smooth surface. It is more like a rough surface here. This is a structure of a lipid bilayer which is the part of the cellular membrane, and this membrane actually contains lot of proteins. You have a glycoproteins which is part of this cellular membranes. You have the transmembrane channel protein. What is meant by transmembrane channel protein? Like this protein is actually part of one cell as well as the part of the neighboring cell. So like it is shared between two neighboring cells, that's why it's called transmembrane cell protein. Then you have the inner membrane protein, and you have this is called the lipid bilayer. Bilayer means, as the name suggests, it has two layers. It is composed of two layers. One is the top layer, one is the bottom layer, and each layer contains a number of different type of protein molecules which they contain. Mitochondria, it is called the energy house of the cell. Now you have the outer membrane which is essentially used for lipid synthesis or the fatty acid metabolism. You have the inner membrane, so it is a two membrane again structure. So one is outer membrane, one is the inner membrane. Now inner membrane actually is useful for respiratory chain or ATP production. You have the matrix here, this is used for the TCA cycle, and you have the intermembranous space which is used for the ADP to ATP conversion. ATP means adenosine diphosphate to adenosine triphosphate. That conversion, that releases the energy and which is used by the cell, right? So this ADP to ATP conversion takes place only in the intermembranous space. So that is the region where this energy is produced. Now endoplasmic reticulum and the Golgi apparatus, this view graph shows their actual morphology. Now these entire things which is very near to this cell, this is called endoplasmic reticulum. You have some rough endoplasmic reticulum, that is ER, and you have some smooth endoplasmic reticulum. Then you have the Golgi apparatus, which is also kind of a interconnected layer like structure, and you have the entry phase, you have the exit phase of the Golgi apparatus. You have the structure of the mitochondrion, which is very clearly shown here. This is a TM image, and this TM image essentially tells you what is the typical dimension. This micron bar is kind of 100 nanometer. That means this length would be roughly around 1 micron. So this is the 1 micron, that is the typical size of the mitochondria. Sometimes this size is also important, because this size will tells you to identify, when you look at the cell structure, that how fine or how small this mitochondria would be in real life, right? And then inside this mitochondria, you see this is the inner membrane space, that where this ADP2, so this is the between these two membrane where the ADP to ATP synthesis takes place, and that releases energy. And this is actually reflected here in this series of the parallel kind of or concentric kind of lines here in the mitochondrial structure. Now examples of the different cell types. Now here I am mentioning that what are the different cell types that are possible. Now if you start with the blood cells, you have the RBC, which is called erythrocytes. You have the lymphocytes, which is the immune system, and you have the monocytes or macrophages. In the connective tissue, the first cell lines which is important is the fibroblast. You have the osteoblast. Osteo means bone. Bone forming cells is known as osteoblast. Chondrocytes, that is the cartilage tissue cells. So cartilage tissue cells are known as chondrocytes. Now you have the endothelial cells, that is called blood vessels. Okay. Now you have the neurons, which is part of the nervous tissue. You have the epithelia, that is either secretory cells or absorptive type of cells. You have the muscle, that is skeletal, cardiac or smooth muscle cells, and you have the sensory cells like hair cells or rod cells, etc. So out of these, what are the important thing for you to remember? Like erythrocytes, here it is called fibroblast, the connective tissue cells, osteoblast cell lines, chondrocyte cell lines, endothelial cell lines, and neuron cell lines. Because in biology you will experience or you will see n number of terminology, n number of different cell types. It is not easy to remember for a non-biologist to really remember all the different cell types. So whatever important cell types that you would experience in your research or in your study, that is enough, and that I have marked it by this star. Again I repeat, fibroblast cell lines, osteoblast cell lines, chondrocyte cells, endothelial cells, erythrocytes, monocytes, these cell lines are important for you to remember. Now again here these major tissue types like endothelial, epithelial tissue, connective tissue, muscular tissue, these are important, and these things I have mentioned here. So epithelial tissue provides covering, and these are characterized by cell shape and number of layers. Connective tissue actually provides support. So, and this support system actually, that means it can offer mechanical support, for example bones and ligaments. It can originate from the mesoderm, that is middle layer of the embryo, and muscular tissue, it is responsible for movement like skeletal, cardiac or smooth muscle cells. So epithelial tissue actually is like a forms like a protection from the outer environment. Connective tissue, it offers you mechanical support. Muscular tissue, it offers you like skeletal or cardiac tissue, and nervous tissue, it provides you control, like total control, like the way you move, way you run, everything is controlled by your nervous systems, or it provides the control of the body functions. Now what is the typical cell numbers on human tissues? Sometimes these numbers are important so that you have a feeling that what are the typical cell densities. Cell densities can be 1 to 3 billion, like 10 to the power 9 cells per milliliter. That is the typical cell densities you have. Now the volume of a 50 kg human is like 50,000 milliliter. Okay. Now 50,000 milliliter. Now if 1 milliliter contains, let's say you talk about 3 into 10 to the power 9 cell lines, and you have 50,000 milliliter. So that is again 5 into 10 to the power 4. So total you have roughly equals 15 into 10 to the power 13. That is the number of cells that you have in the typical human body. So when you talk about cells, now these 15 into 10 to the power 13 cells, they are of all different types. Like you have some connective tissue cells, you have some osteoblast cells, and so on. So around 10 to the power 12 and 10 to the power 13 number of cells are there in the human body. It also depends on what is the weight of a human body, and accordingly total number of cells also will be changed. So roughly for a 50 kg weight human body, you have roughly around 10 to the power 13 number of cells. Now this shows a typically more kind of very schematic picture of the cells, and here it shows again clearly that what is the three important components of the cytoplasm, that is our cytoskeleton, that is actin filament, microtubule, and there is an intermediate filament, and all other typical most important cellular organs like Golgi apparatus as well as mitochondria, everything they are mentioned here. In the bottom part of the slide, what you are seeing is the lipid bilayer structure, and this lipid bilayer structure contains, one is the integral protein or peripheral protein, and then you have the cholesterol here, and then third one is that you have the hydrophobic core. So this is the hydrophobic core region. You have one layer of protein here, you have another layer of protein here, and that is why you have these different lipid bilayer proteins. This is what earlier I have told you about the nuclear membrane, or nuclear membrane, what is the typical structure. Again, this membrane also has a typical bilayer type of structure. You remember mitochondria also have a bilayer structure, and also some of the other materials also has a bilayer structure. In the nuclear membrane you have the outer envelope and you have the inner envelope, and you can see the small pores here. These small pores are actually form nuclear pore complex, NPC, and this NPC, through these pore channels your proteins can be transported to and from the nucleus. So either the protein can be transported to the nucleus or protein can be transported out of the nucleus. So this internal structure or biological structure of the different cellular organelles are important for you to understand the different kind of mechanisms that you will see in the future slides, that so that you will understand that, and you can correlate how they can be correlated with the internal structure of the cellular organelles. So remember, nucleus, just like mitochondria, also has a bilayer membrane, and you have an outer layer membrane and you have an inner layer membrane, and inner layer of the nucleus contains the nuclear pore complex. That means a finely divided pores, it is there distributed in the nuclear pore. The other thing that is important to understand, that you know that inside the cell or from one cell to another cell, the protein molecules, they are transported throughout the living human system. Now how these protein molecules they are transported? For example, if you have a lipid bilayer, okay, and in this lipid bilayer all these protein molecules are present. Once these protein molecules are in critical number, then it form a vesicle, and this small vesicle can be transported and it can be attached to the another cellular organelles, and so that it can be transported to the inside the another cell. So essentially this can go to the from the donor compartment to the target compartment. Donor can be one cell type, target can be another cell type. So from one cell to protein to another cell protein, that is possible biologically. Now what is the chemistry of the cytoskeleton? Typically they have shown here one for mammalian cells like fibroblasts or osteoblast cells, and one is the E. coli bacterium. Now water content is around 70%. So all the cytoskeleton is essentially a water rich environment. Then what is important here, the protein content here 15 to 18%, and then rest is you have the polysaccharides or phospholipids or DNA or RNA. So in case of the E. coli bacteria, your DNA and RNA are all there in the cytoplasm, but in case of the mammalian cells, your DNA and RNA are mostly enclosed in the nucleus. So essentially you have the water, and this water plus proteins, this is essentially the major constituents, and they make like 85 to 88% of the total cell volume. Rest is that either DNA, RNA or some phospholipids and small metabolites. So that's it. that is the content for the cytoskeleton. Now this is like a chemistry of a bacterial cell. Here you have the 70% water and 30% chemicals. Now 30% chemicals means out of that you have some ions, you have some DNA and you have some RNA. So RNA is 6% and DNA is 1%, and you have protein content is the majority, or 50% of the other chemicals is protein, and that is means, so protein percent is 15%, and you have polysaccharides are 2%. Now all these proteins, DNA, RNA, they are called as macromolecules. So essentially these are like polymeric materials, and polymers means it is like number of Mar units, they are coming together and they form this macromolecular structure. This actually clearly shows, this view graph clearly shows that what is the typical structure of the cytoskeleton. Now cytoskeleton, if you remember that, you know, it is a very woven neat kind of formation. It's like a net. You have seen the net. In the net you have this, all this either square type of net or circular type of net. So this kind of, it is like a fibrous structure like actin filaments, intermediate filaments and microtubules, and this fibrous structure, they are also interconnected, or they are like crossed fibrous structure. And this kind of fibrous arrangement, any kind of physical object which is a fibrous arrangement, they can support or they can bear lot of load. So that is the reason this kind of cytoskeleton structure, they essentially provides mechanical support. So mechanical support, that is possible because cytoskeletons essentially you have a extremely fibers and well knit, well interconnected kind of structure. This is the typical arrangement of the actin filaments. You can see it is like a long chain like molecules, like a macromolecular structure, and this is here it is shown like how the different Mar units, they are connected to each other and they are attached to each other. And here it shows that how inside the cytoplasm, how these actin filaments, they really form a very close knit fibrous like structure which is distributed uniformly almost throughout the cell surface. And if you see that more relatively description of this actin filament, it has a more like helical type of arrangements. And if you look at the dimension of this actin filaments, if it is 25 nanometer, so each long chain can be somewhere around 200 nanometer. 200 nanometer means it is roughly around 0.2 micrometer. So that is the typical length of this actin filament structure. And what is the typical mechanical properties of the actin filament? So here it is the deformed force and here is the deformation. And what you see that actin filament, it has a more linear type of behavior, like a brittle behavior. Microtubules also has a more linear type of behavior, it goes to fracture. And intermediate filaments has a more initially linear and then non-linear behavior. So non-linear behavior or more strain to fracture, that is possible in the intermediate filaments, but all the actin filaments and microtubules, they have a exactly like brittle behavior, and which is more like a classical brittle behavior, classical linear type of behavior. I think I'll stop here, and in the next lecture I'll continue with this different cell properties.